Good morning, everybody. Like I said, I'm Richard Turner. I'll be talking about stepwise evaluations of high technology production systems. It says by omission, but I decided to do it by addition, research techniques. As I said, I'm Richard Turner, and I work at Silent Shade Planting Company in Belzona, Mississippi, and I am the lead agronomist. We farm about 12,000 acres, all in Humphreys County, and there's my email contact if anybody needs to get in touch with me. But I did my master's and my PhD research at the Stoneville Delta Research and Extension Center. And this will be pretty much the summation of all of my research that I did for my PhD. So just the main principle of this talk is going to be the evaluation of the economics on the management systems and the production system. And in my dissertation, this will be a combination of chapters 3s and chapter 5. And I'll go through and talk about some of that kind of stuff. But for this presentation, this will be some of the research that we looked at that was done at Stoneville and some that was done at Verona or the North Mississippi Research and Extension Center. So just some of my dissertation format. Chapter 1 was my lit review. Chapter 2 was the interaction of row configuration and seeding rates for irrigated soybeans, and that had 2 planting dates. Chapter 3 was the evaluation of soybean production systems. 4 was the evaluation of the systems, but what we were going to do is we were going to look at how those systems impacted soybean seed composition. 5, kind of the same thing. It's a representation of chapter 3, but at a different location, and that different location was under rain-fed conditions, so it was not irrigated. And then chapter 6 was we were looking at the impact or the differences in non-symptomatic soybean seed compared to infected soybean seed that had purple seed stain. But for this presentation, I will be mainly focusing on chapter 3's and chapter 5 data. So the primary objective was to determine through stepwise addition how those additions impacted agronomics and the economic production side of the soybeans. Like I said, chapter 3, we also wanted to see how those management systems interacted in both irrigated and in non-irrigated situations. And we wanted to be able to determine optimal planting densities for single row and twin row in both systems planted at different timings, and how those treatments impacted seed quality. And then again, like I said, to compare the purple seed stain. And like I said, I won't be getting into all these objectives that we looked at. I'll mainly just be focusing on the first and the second tier that we have there. And then to determine how those impacted several factors from time to time and from season to season. Because like I said, for our PhD research, that was over 3 years. So to be able to see how something, responded or reacted over 3 years, 3 different environments, I mean, that has a lot of power and a lot of importance. So some of our materials and methods at Stoneville, we use the credens, the 4748 Liberty Link. We use that in all years of our study. At Verona in 2018, we used the credens 4748, but we had some difficulties with some herbicide movement. So we moved to a Pioneer 48 Extend technology for the 2019. Soil textures, they were pretty similar across both sites, but you can see we had a Commerce Silt Clay Loam at DREC, and we had a Leaper Silty Clay Loam at the North Mississippi Research Extension Center. The layout of our design was a split plot design, row configuration by planting population, and then our subplot was our actual management strategies placed on top of our whole plot design. We had 2 site years each year, so in theory, we were going to have an addition and an omission study, but later on, we just combined those. We wanted to do a stepwise evaluation of components, and the irrigated location site was at Stoneville, and the rain-fed irrigated site was at Verona. So this is just going to be a visual representation of some of the row configuration that we had. So at Stoneville, and you can see at the top there, this is for DREC, we are on the 40-inch bed system, and we have single row and twin row, and this is a representation of each. The top picture on the right is going to be the single row, how that's laid out, and the bottom picture is how the twin row system is laid out. Planting populations for this study will be designated medium and extra high, and those seeding rates, designated seeding rates for each of those denotions. That was determined on um, the amount of seeds that are planted per foot. So medium comes out to 8 seeds per foot, and extra high comes out to 12 seeds per foot. And this is how our addition of our subplots were laid out. So our none would be our control. F only is just going to be a fungicide applied at R4. And each one is a stair step. So it's adding additional management strategies. So it's the fungicide plus the desiccant, fungicide desiccant plus additional P and K. So don't forget there, that's additional P and K. So no soils were limiting for their designated fertility, but we wanted to see the impact of what happens if you applied additional fertility. And then the same thing for the fifth one, we added nitrogen sulfur in the form of AMS for additional nitrogen and additional sulfur to see how that impacted soybean yield. So same layout for the North Mississippi Research and Extension Center, but because they're on 38-inch beds, it did change slightly, mainly because we changed the row orientation, but we wanted to keep the same 8 and 12 seeds per foot. So because of the row configuration, the row width differences, that actually made the 6 and the 12 change population slightly, and that's denoted there. I'm going to keep them the same, medium and extra high, but just do be aware that that actual population per acre did change slightly. But the subplot, the additional, the management strategies, they did not change. So some of the measurements that we took with Stan, we took those early and late. We wanted to know how many seeds we were losing or plants we were losing during the growing season. We took canopy closure measurements that was taken using a cell phone app called Canopio, and we did those from R1 to R7. We did grain index. So at harvest, we captured a sample, a sub sample, and we were able to do seed weights, bushel test weight, grain moisture, all that kind of stuff. The oil and protein content, that was done in cooperation with the USDA and Dr. Nasser Bellalui. And we already have a publication out of that chapter, but I will not be discussing those results in this presentation. We did a partial budget, but that was strictly going to be based on planter equipment, seed costs, and cost management strategy. That had no herbicide or insecticide inputs and factored into that. We did a percent purple seed stain infection, and then the effect of purple seed stain on seedling composition or seed composition, seedling vigor, and germination. And like I said, we won't be talking about that in this presentation either. So the first bit of data that I'll get into, and this was done using our Canopio app, and this is comparing all the different management strategies, as I mentioned earlier, with the control, fungicide plus desiccant plus additional P and K, and again with the nitrogen and sulfur. And as you can see from this graph here, management strategies didn't really have a huge impact on our the effect of canopy closure. That's not really surprising, because as I flip over and get into the next slide, you can see very much so that canopy closure was much more greatly impacted by row configuration and our seeding rates. I am aware that there's extra seeding rates in this one, and that was done from another chapter, but I did think that it helped. I thought that it helped show just the importance of how planting population can have a big factor. So you can see that the extremes here are going to be made up of the wide bars, the blue and the red, and that's going to be the row configuration. So single row had much less canopy closure when compared to twin row. And the same thing happened to our medium and extra high populations. The extra high population had a much greater canopy closure early in the season. Why is early in the season important? So most of our herbicides that we actually can use sprayed over the top, most of those cutoffs are going to be at that R1 time frame. So having the most canopy closure possible at that R1 time frame is very, very essential, because essentially at that R1 growth stage, we don't have as many options to go back in and clean up messes if we were to have weed slips or something of that nature. So The further along in the growing season that we got, actually the margin of error, they started merging. The single row, twin row got a lot closer, and as well as the planting populations. So they didn't impact canopy closure later in the growing season, but as I stated earlier, that R1 growth stage is critical. So seed index, this is going to be seed weight. And I have it here for Stoneville being the black bars and Verona being the maroon bars and their level of significance. So with the control, you can see both sites was considerably less than once we started adding management onto that. So essentially what that means is we were getting heavier seed with the addition of the fungicide. And that can be seen at Verona and Stoneville. But you can see a slight numerical drop off starting to occur. We're not really sure exactly why that happened, especially when you get into the fertility. What makes the fertility make the seeds weight or the seed index start to deteriorate? And we really never really got a good grasp on that, and never really knew why that was beginning to happen. So moving on into purple seed stain. So this is a four-way chart if you want to think about it like that. So the black bars will be the row orientation just at Stoneville. The maroon bars will be the row orientation just at Verona. And then moving over to the right side of the graph, the gray bars will be Stoneville. So I kind of tried to keep them color coordinated. So red, maroon is all Verona, gray and black would be all Stoneville. But even though we see, differences in bar heights and bar levels here, the only significant interaction that we had with a whole plot impact or interaction, or not interaction, but response was going to be twin row reduced purple seed stain when compared to single row only at Stoneville. So we didn't see it happen or occur at any of the other locations or by seeding rate. So it was just a whole plot row orientation effect. However, purple seed stain was greatly impacted by our management strategies. So again, you can see the C bar being our highest at most all, at across both sites, being Stoneville and Verona. And with the addition of the fungicide, you see the level of purple seed stain infection come down. So we are getting, you know, some control of that disease or of that bacteria that is causing the purple seed stain. So by having that fungicide out there, we are actually able to reduce the amount of purple seed stain. And it came down with desiccant because, and we have a feeling that with the addition of the desiccant, we were able to actually get in and make a lot of the harvest in a more timely manner. But really, once we added the desiccant, we see that the separation really began to drop off, and we didn't really see a whole lot of differences beyond the addition of the desiccant. So grain yield, again, same thing. At both sites, at Verona and Stoneville, we saw a significant increase with the twin row system as compared to the single row system. We have a lot of, we do believe that has a lot to do with, competition, more airflow, lots of things are happening in that twin row system as compared to the single row system. But if you'll look over on the right hand side of the column, um, we also have, we see where the extra high seeding rate actually increased soybean grain yield too. However, some things that come into more seed is going to be added cost. Seed is a great expense that we have. But at both Verona and Stoneville, we had the situation to where extra high or the 150-ish thousand seeding rate outperformed the 108, 110,000 seeding rate. So getting into our yield with our management strategies, we did see a slight increase by going from no management to a fungicide only. Again, that could be, were we saving yield because of diseases? Were we just protecting yield? We don't really know the answer to that question, but we do see where the increased fungicide is increasing soybean grain yield. But we do have, again, that kind of tail off with the addition of the desiccant. And we think what's happening there is just the addition of the desiccant. We actually had header loss of up to 40% greater than that of an untreated or a fungicide only, the amount of seeds that were lost at the header. So some of that yield decline is due to header loss, but we don't think that that's all of it. Might be some seed weight or something of that nature starting to tear off as well that we saw in seed index. But a keynote to take away from here is the use of additional fertilizer never increased grain yield. So that's an important takeaway note here. It is just because if it's not limiting, it's not making you more money putting it out there at this one given time. So it won't make you more yield this year. So in return, and obviously this isn't a very big shock, the control and the fungicide only, that they were pretty similar across the stage, across all years, and averaged together. But with the addition of the desiccant and the fertility, we see the return per acre start to tear down, because none of those products or none of those systems were making you money. But don't think that I'm saying don't use desiccant, because I am very much so aware that the use of a desiccant can allow producers to get in and harvest a field in a much more timely manner if we have bad weather approaching or anything of that nature. So I'm not discouraging the use of any of these practices. I'm just saying that it's not something that will make you money. But like I said, if it allows you to get a earlier harvest done, it very much so can. So take that for a grain of salt. So the take home message of my presentation and all my research is scout. So weed control is essential. So go out, make sure you have weeds. Don't make unnecessary applications. Row configuration can lead to more yield, and it also can lead to reduced inputs in some kind of way, such as weed control. So not only can it make you more yield, but it can also save you money if you don't have to make as many applications. Seeds are expensive, so keep that in the back of your head. Don't make unnecessary replant decisions if that's not if we went out and planted 150,000 seed, but we're still in that 100,000 seed range or plant range, don't do a replant. So we have plenty of data that shows that 100,000 seed can make competitive yields. So definitely don't go out and replant. Fungicides can increase yield and reduce the presence of purple seed stain, but it does have the additional cost that's associated with an aerial application and the cost of the product. And some of those products can be expensive. Fertilizer is essential. So I'm not saying fertilizer isn't important. Fertilizer is very much so important, but increased rates or excessive rates don't make you more yield. So the take home message from there is do a $6 soil sample and apply what's necessary. Do not apply more unless you're trying to build your soils up to get them into a higher category to where maybe you could prevent or not have to do as much fertility in the future. So with that, this will wrap up my presentation. I do have here the two two papers that we already have, journals that we already have printed. These are on the left-hand side here, along with the names and what journals they're in. And I would like to thank the Mississippi Soybean Promotion Board and Dr. Ebelhair for being my major advisor and for funding my project. And at this time, I will take any questions if we have time. If not, we'll be moving to the next presentation, and I will stop sharing and again drop any questions in the chat box.
Excellent, thank you. Yeah, let's go ahead and move on, but we do encourage everyone, as you have questions, to put them in the chat, and we will address those as time allows. Dr. Popescu, we'll let you take it over.
Thank you, Michael. Are you able to see my screen? Is everybody okay? All right, that everything looks okay? All right, so my name is Serena Popescu. I'm an associate professor in the Department of Biochemistry, Molecular Biology, Plant Pathology, and Entomology here at Mississippi State. I'm very grateful to be able to present our results on this forum this morning. And I want to tell you about what we've done on a project that we started about a year ago regarding root microbial communities and their role in helping us grow healthy and disease-free soybean. So I'd like to start with a short background on rhizome microbiomes. So define rhizome microbiomes. So what are they? So basically, as plants grow, they start, they start interacting with microbes. The roots start interacting with microbes from the surrounding soil. And some of these microbes start growing on the surface of the roots, as you see in this picture. And we call these communities the rhizosphere communities. Other microbes are able to grow inside the superficial tissues of the roots. And you see in this diagram here, these tiny red dots, these are microbes that are able to go inside the roots and start establishing and growing inside the plant. We call these endophytes. So together, the rhizosphere and the endophytes compose the rhizome microbiomes or root microbiomes or rhizome microbiome communities. So many names for the same thing, basically. So the microbes that are interacting with plants can be fungi, can be bacteria, oomycetes, protists, so all kinds of microbes growing in very dense communities associated with plant roots. So how are these communities established? So why does plant interact with microbes? Well, there is a very complex molecular communication going on between plants and microbes. So on the one hand, plants secrete chemicals in the soil. This can be sugars, amino acids, and these chemicals are seen as nutrients, are used as nutrients by soil microorganisms. So they're attracted to the plant roots and they start attaching themselves to the roots or going inside the plant. And themselves, these microbes are able to secrete certain chemicals that plants are using to fight pathogens or grow healthier or fighting all kinds of stresses from the environment. So this research that has been going on rhizome microbiome is at the basis of the so-called biological control. It's an alternative way to control pathogens and diseases, and at the basis of the so-called microbe-optimized crops. So breeding microbe-optimized crops. So these are crop plants that are able to establish interactions with specific beneficial microbes. So why is it important to understand rhizome microbiomes? Well, because they can help us fight a lot of these very destructive pathogens that are attacking soybean crops. So one of these pathogens is Xylleria. And Xylleria, it's an emerging pathogen and it's the cause of the taproot decline in soybean or TDS. So what you see in these images right here are symptoms of plants that are infected by Xylleria. So chlorosis of the lower leaves in the canopy is one of the earliest symptoms of infection. You can see sometimes fungal stroma growing in the root of the soybean plant. Fungal mycelium present in the soil around the roots, and eventually the roots become black. And in extreme cases, the plants die. And this is how Xylleria looks when you grow it in the lab on a Petri dish. It has this blackened appearance with white mycelium. So TDS, it's an emerging disease, has been reported not only in Mississippi, but a lot of the southern states that are growing soybean. And it's an economically significant disease to soybean production. And I want to acknowledge here the work that has been done at Mississippi State by research faculty and extension faculty on this pathogen and characterizing this disease. Work done by Dr. Tommaso Peterson, Dr. Allen, Dr. Wilkinson, and others. So we started working on this project about a year ago, as I mentioned, and we had two main objectives here. So we wanted to understand what is the composition or structure of root microbial communities in Mississippi Delta soybean, and then identify microbes with the potential to control TDS and Zalaria and help us grow healthier plants. And we started this project with a very targeted question, with a pilot project in which we basically asked, are there any endophytes? So those microbes that go inside the root of the plants, are there any endophytes that have been isolated already that may have antagonistic activity against Zalaria? And what I did, I basically got in touch with some of my colleagues at the University of North Carolina, Chapel Hill, a lab which has been working for a long time on root microbial communities, but they've been working on the root communities isolated from this tiny plant. It's called Arabidopsis thaliana. It's a weed actually, but is a system, a model system for plant biologists. So this lab has isolated thousands of endophytes from the roots of Arabidopsis, and I was able to go through their library and select about 30 strains that, based on literature and based on my own judgment, I consider to have a potential antagonistic activity against a fungal pathogen. And out of these 30 strains that we were able to get from UNC, about 21 actually grew on the plates in our hands in the lab. So we decided to test these strains for their antagonistic activity against Zalaria by using these so-called co-cultivation assays, where we basically have Petri dishes where we grow Zalaria at one end of the Petri dish and the endophyte at the other end. And then we compare the growth of colonies, Zalaria colony on these plates with the control plates where we only grow Zalaria. So this is how real life plates look. So this is a control plate with Zalaria growing happily at this end. And then these are plates with two different endophytic strains and Zalaria growing, as you see, lower with a lower diameter colonies. And this is basically a statistical analysis of the results from the 21 strains that we tested from this library. What you see on this axis is the inhibition, percent of inhibition. The lower the number, the higher the inhibition of Zalaria growth. And you can see about 11 of these strains had a significant effect on the growth of Zalaria on the plate, with the others showing either not significant or no effect on the growth of the fungus. So this was very encouraging, actually. So we took the next step and decided to start building our own libraries and start really work on the soybean microbial communities. So these are some images from last summer when we went to the Delta. We visited a few farms, a few fields, and we got samples from one of the more heavily Zalaria infested fields that we found. We got samples from healthy soybean plants, but also samples from plants that were showing mild symptoms of Zalaria infection, such as this chlorosis that I mentioned, and also plants that were heavily infested by Zalaria, almost dead, basically. So we had healthy plant samples, mild symptomatic samples, and heavily infested soybean samples. So we got these plants to the lab, back to the lab, and we basically started two sets of experiments as shown on this slide. So in one set of experiments, we extracted DNA from the healthy roots and infested roots, and then we sent them to a company for sequencing. So basically what we've done with that DNA was to sequence the genomes of all the microbes that are present on healthy and infested plants with the idea of performing a meta-taxonomy and a very thorough comparative characterization of the microbiomes of healthy and Zalaria infected plants. In the other set of experiments shown here at the bottom, we extracted the root microbes only from the healthy plants. We cultivated them on very many types of lab media with a goal of isolating single colonies and making our own root endophyte library to start testing them for potential beneficial activity. So let me show you now some of the results that we got from this first set of experiments on metagenomics. So what you see in this graph is basically fungal families, the way they were identified by the metagenomic analysis in the healthy plants, plants with mild symptoms and plants with heavy symptoms. And all these colors represent a specific fungal family. So you see that some of these fungal families are present in all these type of plants. Others are present only in the healthy plants. Others start accumulating in the diseased plants, in the infected plants, such as, for example, of course, as we expected, Xyleria was heavily abundant in the mild symptoms, heavy symptoms plants. And when we look at Xyleria strain, what kind of strains we have represented in these plants, you see that healthy plants have no Xyleria infection, but mild and heavy symptomatics now are heavily colonized by Xyleria, with certain strains present in these particular populations. And we also see that not only Xyleria C, but also other types of fungi, such as the ones from Magna Portaceae family. So this is a family that has a lot of pathogenic type fungi, and we see it very abundant in symptomatic and heavy symptoms plant, but not as much in the healthy plants. These are some of the strains we identified. So this is how the fungal populations look. How about the bacterial populations? So again, looking at healthy, mild, and heavy symptomatic plants, we're looking at classes of bacteria that are present in these microbiomes. Beta proteobacteria were some of those bacteria that accumulated heavily in the infested plants, with a lot of species heavily abundant in mildly symptomatic plants. Gamma proteobacteria, the same situation, with nothing in the healthy, but abundant in the infected plants. And they were more abundant actually in the heavy symptoms plants. Looking at some of the other classes, other families of bacteria, so Bradyrhizobiaceae. So these are nitrogen fixation bacteria, which are very useful, of course, for the plant because it helps us fix nitrogen and grow healthy. They are predominant in the healthy plants, but in the infested plants, you see that their abundance decreases and other families come up. Some of these Bradyrhizobiaceae strains are useful strains in nitrogen fixation, but some others, such as this Elkanii strain, is a known pathogen from this family, which was shown a long time ago to cause leaf chlorosis. So one of the big surprises that we had looking at these communities and comparative analysis so far was that not only Xyleria starts, infecting the plants and accumulating in the mild symptoms, heavy symptoms plants, but a lot of other bacteria and fungi with pathogenicity and virulence factors. So looking at just the virulence factors, those strains that are able to cause disease in plants, you see there's almost no virulence factors in healthy, but look at the heavy symptoms plants, how heavily colonized they are by these pathogenic bacterial families, meaning that basically Xyleria infection sort of opens the gates for all these other strains of pathogens to come and infect the plant. All right, so let me now switch and tell you about the second set of experiments, making the library and testing these pathogenic strains. So so far, we were able to isolate about 230 distinct bacterial strains from healthy soybean roots. we tested and we have results from about 1/3 of this number, about 70. We are currently testing about 1/3. We don't have the results yet, but soon I'll have the results from these strains as well. And we hope to finish up testing this library by the summer, maybe end of the summer. So what we see in what we've tested so far, the majority of them had no effect on Zalaria growth, but we had a number of these strains that have a significant effect on Zalaria growth. And what you're seeing here is basically those co-cultivation assays where we grow Zalaria on the same plate with these isolates. So a few of them have a very, very strong inhibitory activity on Zalaria growth, some moderate, and a few low but still significant inhibitory activity. These have no effect. And surprisingly, we also see a few strains that actually increased Zalaria growth on the plate. So they augmented the growth of this fungus on the lab media. So you may ask at this point, yes, you see this beneficial activity, in the lab on these plates. What happens with the plants in the real, sort of a real world situation? So we've designed an experiment to basically test whether these beneficial strains that we identify in the lab on these plates have an effect on taproot decline that we can cause in a soybean plant. So this diagram shows you the experiment that we put together and optimized. So we take soybean seeds and we cover them with, so we do seed treatment with each of these beneficial strains that we identified in vitro. And then we plant them and let them grow to about cotyledon stage. And then we infect the soil with a plug containing Zalaria mycelium. And we have controls in which we do not treat the seeds, we infect or not infect, or treat the seeds but not infect with Zalaria. So all these controls, they grow together in the growth chamber. And at 4 weeks, we take the canopy, we dry it, and we calculate the dry weight and look to see whether our strains have an impact on Zalaria infection. So this is what we've have so far in terms of results. Let me show you first the controls here. So these bars show, these are the control plants. So this is a plant grown just like here, no infection, no coating with bacteria. This is a plant that has been infected with Zalaria. So it's this situation here, but not coated with any isolate. So comparatively, you see this big impact on Zalaria infection, which of course it's quite obvious when you look at the picture. These plants eventually die, it's about 60% inhibition in canopy dry weight between non-infected and infected plants. All these other plants have been coated with these endophytes, and looking at them, except this one as well. So looking at them, you see that basically, so the black bars are the ones that were not infected with Zalaria. These are plants that were coated but infected with Zalaria. So basically, you see no statistical differences between infected, not infected plants. Basically meaning that if we coat the seeds of these plants with this particular endophytic isolates, we obtain about 100% protection against taproot decline. Another interesting thing that we observed was that one of these strains actually helped the plants grow taller and larger leaves, so significantly larger than the control plants. So we see some growth enhancement activity for this particular isolate. For these 4 isolates, we do not see that growth enhancement activity compared to these controls, but they do have about 100% protection against Zalaria and taproot decline. For these 3 strains, we see 100% protection against Zalaria, but we also see that by coating the seeds with these strains, we actually inhibit a bit the growth of plants that were not infected with Zalaria. And this was our negative control, one of the strains which we tested in vitro and showed no protection activity, and we don't see that protection activity also when we do this type of experiments on the plants. All right, so this is very encouraging, and we are continuing testing all the strains that are coming out from the in vitro experiments. We are continuing with the analysis of metagenomes and doing metataxonomy, continuing with screening of the isolates, both in the co-cultivation assays and on the plants. And at the moment, we are actually sequencing to identify these beneficial isolates. So we want to see what strains these particular isolates are from. Is there anything specific about these particular bacterial strains that make them to have these beneficial effects? What do we want to do next? Well, we want to see how broad is this antifungal activity that we found. We want to test against other root pathogens such as Macrophomina and Rhizoctonia, both particularly heavy pathogens. We want to find out what is the nature of this antifungal activity. Can we extract the active compounds from these bacteria? And we started some work on this particular goal. And we want to find out whether this beneficial activity is stackable. Can we combine these beneficial strains in certain combinations so that we get growth enhancement and protection against fungal infection? Maybe not only against Alaria, but maybe multiple pathogen protection. So these are some of our goals for the coming year. And I want to end by acknowledging everybody that's been working and helping with this project, starting with my two wonderful co-PIs, Maria and Teresa. I want to also thank Dr. Allen for his help with selecting samples this summer, George for his help with the analysis of the metagenomics data, producers from the Delta who allowed us to get samples from their farms, all the people actually doing the work on this project, professional, Dr. Aline Bronzato Badial, she's a postdoc in my lab, Setare Jasmine and Maurice and Rizwana, they are graduate students working on this project. And the many undergraduates that have joined us to help bring this project forward, Griffin, Sam, and all the Josh, Aja, Autumn, Trinity, Tiricus, and BK working this semester with the graduate students to screen the library and the plants for beneficial activities. So, and thank you all for listening, and I can take questions. Thank you.
Yeah, I think there are a few questions for you in the chat, but we'll let you maybe just address those as we go ahead and move on in our presentations, and you can address those one-on-one. Is that okay?
Yes, yeah, thank you.
All right, Dr. Brown, we'll move to you, and if we're still having those little bit of technical issues, Stacy will do the presentation for you, and you can just have her advance your slides.
Sure, I'll go ahead and try.
Okay.
It appears I'm still having the technical difficulty, so Stacy, I apologize, but you're gonna have to share my presentation. Okay, thank you. I'm really excited today to be able to talk to you about some of the work that we've been doing over the past few years. Really sort of enhancing the testing capabilities to be able to detect 2,4-D and dicamba, and also how that can be used in a stewardship program. So there's a lot of people that have been involved in this project. And so I'd like to also thank some of my colleagues and co-PIs on the project as well, listed as authors here. So if you can advance to the next slide. I'm kind of in a unique situation. I serve in a dual role. I'm the state chemist and director of the Mississippi State Chemical Laboratory. And that laboratory is actually the agricultural forensics lab for the state. So our mission is to provide analytical data for quality assurance and labeling, safety of feed and fertilizers, human foods, pesticides, and actually the petroleum products that are sold within the state. We have 4 divisions within our laboratory: our Industrial and Agricultural Services Lab, which is essentially our fee-for-service lab. So we can offer our analytical capabilities, to do a variety of different types of testing for researchers or for producers or for people in the community. And then we have our regulatory division, our petroleum products division, and a research division. So part of the mission of the laboratory is to assist in research that is going to advance the field of regulatory science. And so as the state chemist written into state law, the state chemist has to be tenured in either biochemistry or the Department of Chemistry. And so I'm the first state chemist to ever be tenured in biochemistry, which I'm very proud of. But I also think that it states to sort of the progression of research, and that, the biochemistry department is on the ag side, and it makes sense, since we are the agricultural forensics lab for the state, that I would be tenured and have an active research program within the Department of Biochemistry. So next slide. So we do all sorts of analytical analysis: feed, forage, food, your minerals and inorganics, heavy metals. We test fertilizer, litter, wastewater, drugs of abuse, controlled substances, hormones, antibiotics, toxins, a variety of tests to ensure the safety of the petroleum products sold not only in this state, but also in Alabama. We do environmental and industrial chemical residue testing, pesticide residue, and also formulation testing, and a variety of different hazardous waste and priority pollutants. So we have a lot of analytical capability, and we pride ourselves on being very adaptable to test a multitude of things, to be able to get answers to help the community, to help researchers, and to help protect the consumers. So next slide. We're an ag state, and it makes sense then that, we're here to essentially protect the agricultural industry. And by the industry, we mean the producers, the suppliers, and the consumers. I kind of feel that the industry is all of those things encompassing in one. So that's essentially what the lab is here for. Next slide. We assist in helping the citizens in Mississippi and in other states with pesticide field application through our formulation testing, to ensure that the products that are being sold actually have the correct active ingredients in it, and that there's labeling compliance with those. So we do formulation testing, and that helps with, some of the regulation, and to ensure that the products are what they say they are. Next slide. We're also involved in the litigation of pesticide misuse. Typically, that's approximately 400 cases a year, give or take, depending on the years. This can be intentional, off-target movement cases, pesticide misuse cases, or improper application cases. So a variety of different cases when we're talking about pesticide misuse. Next slide. So I said earlier that we do a variety of different types of testing and have a lot of analytical capability. But in 2017, I was approached, and I think many of you have probably seen this probably exact figure or similar figures in looking at off-target movement and dicamba drift damage that was seen within the 2017 growing season. And one of the things that I was approached with was, in these drift events, when inspectors are bringing, or producers are bringing, or consumers are bringing samples to the lab, can I detect the formulation of the pesticide that has been applied to that plant? We do formulation testing, but could we actually detect the formulation of what was being applied to the plant? And my answer was no, which I don't like to say. I like to be extremely helpful, but my answer was no. The common regulatory techniques that we use, and I'll go through some of those in just a second, did not allow from extracting those small levels of specifically dicamba or 2,4-D, these auxin herbicides within the plant. When that formulation has been applied to the plant does not really care the specifics of the formulation, those small molecular groups that the individual companies put in to make their formulation their own. The plant is using the parent compound. And so when we're going and extracting that out, those small molecules that make the formulation indicative of the formulation become very difficult for us to define. So that got us thinking, could we perhaps identify the formulations in a drift event? So next slide. So just to highlight, these are some of the formulations. If you look at dicamba and you look at the structure of dicamba in the acid form, you can see that there are these small molecules that I was talking about that are indicative of the individual formulation. So the diethylamine, the dicholamine with potassium acetate, or just the DGA, the dicholamine. Or if you see the BATMA group with a little maroon sort of box around it, this is actually the group that was put into the formulation, which was supposed to help with the volatility and make this dicamba formulation have a lower volatility and therefore help with some of that off-target movement. So essentially when I was asked, it was, can I determine if the correct formulation and use has been used in these off-target movement sites? So was it actually the formulation with the BATMA that was applied to that soybean field and then it drifted over to another field? Or was it one of the older formulations that sort of has this lower volatility and therefore there was an improper use of the pesticide? So next slide. What makes it even more challenging when being asked, could we detect the formulation, is these are auxin herbicides, meaning that when they're absorbed in, they act like hormones within the plant. So they're metabolized. And so if you look at the parent compound of dicamba, once it enters the plant, it's being metabolized to the 5-hydroxydicamba and also the DCSA. And then further metabolized down to the DCGA compound. When we're actually screening for the use of dicamba in particular in the lab, we're not only looking for the parent compound, which depending how many days after application, you may not actually see the parent, but we're also looking for the metabolites. And again, those small molecules that are with the formulation are very difficult to find. There are several amines within the plant system itself. And when we're going through and pulling out these compounds, we're essentially using extraction techniques that are looking and being able to pull out only these types of materials so that we can get a lower level of detection. So I like to say that what I was being asked to do is not even looking for a needle in a haystack. I'm actually being asked to look for a certain piece of hay in the haystack because it mimics what's going on in the plant, and looking for those very small molecules, especially several days after application, can be very challenging. In my years in working at the State Chem Lab, I have never seen a plant sample come through the door that someone has said, I just accidentally applied this and we're going to have a problem. Typically, we wait until we see a problem before that plant has been there. So it's several days after maybe the drift event has occurred, and it's usually at a very, very low concentration. And I hate to say this, but the plants are very good at detecting whether this type of pesticide has been applied to them. In fact, they're a lot more effective than our very sensitive, very expensive analytical instruments are. So a lot of times we know there's been damage, but I can't definitively say because I can't detect it in the instruments. So next slide. So if we look at what we typically do when we're asked to determine if dicamba or 2,4-D or several other pesticides may have been used in a drift event or may have caused damage in a drift event, the plants come in, they're processed, they're ground. We then use some type of extraction procedure to pull out the analytes or the compounds of interest. And when you do that, sometimes chlorophyll and other agents from the plant come over, and so you clean it up so that you can get the lowest level of detection. And then they're analyzed, like I said, on our very sensitive liquid chromatography, mass spectrometry, triple quad instruments where we can get very, very low level of detection. But again, sometimes the plants are even better than our analytical instruments. But this doesn't give us any hope of being able to detect and look at those individual components that make the formulation the formulation. So that got us thinking. Next slide. What if we were to use infrared Fourier transform spectroscopy? And this is similar to stuff that we use in our lab, technology that we use in the lab already. And we were to couple using spectroscopic techniques to chemometrics, to statistical analysis, to build models for detection. If you look at determining nutrient value for feeds and for forages, we use similar type of technology coupled with algorithms to determine and approximate protein, fat, fiber analysis. And of course, we're using near infrared in that case, but we thought, could we use Fourier transform technology, which uses infrared light to scan and test samples, and you look at these sort of surface level chemical changes that might be happening in the plant. And so our thought was maybe these small molecules might play a role in changing some of those chemical bonds, and therefore when the infrared light is exposed to that sample, we might be able to detect through different bending and stretching frequencies and different energy that's associated with those bonds, slightly different chemical compositions that may be indicative of the formulations that were there. And then if we could couple it to software or statistical packages that are similar to facial recognition software. Most all of us have seen facial recognition software, especially if you have an iPhone. Now you don't even have to, you can use your face unless you're of course wearing a mask at this point, but it recognizes your face, which has similar features to just about everybody else's face, but the individual features of your face to say, yes, this is your face, and unlock it. So could we couple these two things together, build models, and then challenge those models to say, yes, this particular formulation was applied to this plant? So next slide. So the chemometric analysis, this sort of looking at the statistical packages, we looked at principal component analysis, and this is a technique which is used to emphasize variation. So it's going to look at strong patterns within a data set, and you can make predictive models using this. And so we're looking at the patterns that are present, and we're looking at the variation within that. If you look at linear discriminant analysis, you're also looking at pattern recognition. You can use machine learning with this. And with this type of model, instead of focusing on the similarities, you're now focusing on the differences. You can even do a joint PCA LDA model, which is going to sort of fine tune the similarities and differences and look for, being able to predict in between those gaps between the similarities and the differences. And it's oftentimes used to predict unknown sample classification. So this was sort of a starting point. Could we couple spectrophotometric analysis to statistical analysis, and could we build models to then predict what type of formulation had been applied? So next slide. So this was sort of the first test that we did. This was sort of ground truthing our project. And so if you look at the first figure, this is actually what the infrared spectra looks like when these auxin herbicides are applied. They don't look very different to the naked eye. So we really have to parse out those very small differences within that. And so what we found when we first ground truthed this is when we took plants that were applied a 1/64x rate of the normal application rate for 2,4-D or dicamba at various formulations, that upwards of about 98% accuracy, we could depict which formulation had been applied to the plants. And so we collected plants immediately after application, 1 day, 3 day, 7 day, and all the way out to 28 days after application. Now, this is a very what I would consider a concentrated application rate for a drift event, but it was very promising that even 28 days after application, we could fairly accurately depict which formulation had been applied. So we then took that one step further, next slide, and we wanted to know what would happen and what would make this more real life if we took out sort of those earlier days of application, the 0 and 1. No one's going to, we're not really going to be testing those types of plants in a real world setting. And so we conducted experiments at the R.R. Foyle Plant Research Center here in Starkville, Mississippi. We did this in both soybean and cotton. And so we used a single cultivar for soybean and a single cultivar for cotton. I will say we are now incorporating different types of cultivars into our models, but the data that I'm going to show was just done with one. We took 6 different concentrations of dicamba and the 2,4-D. So we went all the way from a 1/32x all the way out to a 1/1024x rate for each of those pesticides. And we used 4 different formulations for both the dicamba and the 2,4-D. And again, we extended it beyond the 28 days, and we extended it now out to 56 days after application. So next slide. So I just want to go through a little bit of the materials and methods. Once the samples were collected, the samples were ground. We used about 1 gram of leaf tissue, and it was placed on our horizontal zinc selenium reflectance crystal. That's actually how the light, that infrared light, is passed through the sample. We did it at an incident angle of 60 degrees and 10 reflections per scan. And then we generated the infrared spectra using a Nicolet 6700 FTIR. We automated the baselines and we attenuated those reflections so that they were sort of corrected and sort of normalized. And that was done using the Omnic software as part of the FTIR system. So these corrected spectra were then exported into our chemometric software. In this particular case, we used Unscrambler. And from that, we were able to look at our models or develop our models. And so we didn't pool the cotton and soybean. We did independent data matrices for both types of crops. But what we did do is we took the spectra from the samples treated with all of the concentrations and with all of the formulations and all of the evaluation timings. So looking at the days after treatment. We did that because a lot of times we're not going to know the initial concentration, and we're not going to know perhaps the day after treatment. And so we wanted to see how robust would our model be if we sort of included more variation into our model. And so next slide. So if you look at the classification models for dicamba, for both cotton and soybean, if you look at the types of models that provided the best data, if you look at the raw spectra with the LDA plot for both cotton and soybean, you can see that for cotton we had about 84% accuracy, and for soybeans we had about 79% accuracy. So our accuracy dropped from our sort of benchtop, ground truthing experiment, but still much better than, just having to take a wild stab at it or not being able to determine that. Next slide. If we look at the confusion matrix, and this particular one is for cotton treated with dicamba, you can see that when we took, let's just say, for instance, the BATMA, 90% of the time it was identified correctly. 1% of the time, the BATMA formulation was identified as DGA. 6% of the time, it was identified as DGA with the potassium acetate, and 3% of the time, it was identified as DMA. So for our modeling system for dicamba, with the 4 formulations that we used, we have about an 85% accuracy. Next slide. And if we look at it with soybeans treated with dicamba, again, if you look at sort of the overall accuracy, we have about 80% accuracy for determining the formulations once applied to soybeans, regardless of the timing. So again, these are from pooled concentrations and from pooled sample timings. Next slide. So again, if you look at the classification models for 2,4-D, we see the same thing, that the LDA spectra modeling source provided the best accuracy. Again, 77% overall accuracy for the cotton spectra and 80% accuracy for the soybean spectra. Next slide. And again, if we look at the confusion matrix for predicting the models, you can see that, for instance, for the acid formulation, 73% of the time it was predicted correctly, 3% of the time it was predicted to be DMA, 19% of the time it was depicted to be the choline, and 5% of the time it was depicted to be the ester. So not perfect, but pretty good. Next slide. So in conclusion, our typical methods that we use in the lab, the LCMS, MS methods, cannot differentiate between the formulations. If we look at the joint PDA, PCA, LDA models, they were only capable of classifying 2,4-D and dicamba formulations in damaged tissues between 36 and up to 40%, whereas the LDA alone produced models with accuracy between 77 and 80 and upwards to 85% respectively. So it looks as though this technology using the FTIR spectra coupled to chemometrics for classification, it looks promising, but it does suggest further refining. So some of the things that we're doing now is, we might be able to couple this with the LCMS to say, this is the amount of herbicide that we have. We did see that when we have the known amount and we don't, when we know the concentration and we're not pooling all of the concentrations together, our modeling and accuracy goes up. We do know at the higher concentrations, our models tend to be more accurate than at the lower concentrations. That may not be very good when you're talking about sampling plants that that pesticide has drifted onto, but that may be very good if you're going back and saying it may have drifted from this plot that had the full rate application on it. So we're actually looking at that now, and we're incorporating additional soybean cultivars into that. Oftentimes when we're looking at the actual plot where the formulation has been applied, we're looking at those dicamba and 2,4-D resistant types of technology cultivars. And so we're not seeing the damage, or we wouldn't expect to see the damage. So we're using different models looking at that particular cultivar. And with that, thank you very much. I would like to thank the Soybean Promotion Board for helping to fund this project. And I'd like to thank all of you for listening and for giving me the opportunity to talk about some of our research today. Thank you.
Thank you. That was really interesting. All right, we'll move to our last speaker. Just final pieces of information. Just want to ask everybody to add any questions to the chat if you have any for any of our speakers. Okay, Dr. McCoy, we'll let you take it over.
Okay, I hope everybody can see those slides. So as you said, my name is Justin McCoy. I'm here with Dr. Mary Love Taggart today. Speak a little bit about the effects of cropping systems on iron deficiency chlorosis. Before we begin, I'd like to thank the Soybean Promotion Board for funding this research and also giving us the opportunity to speak to everyone today. So I'm going to talk a little bit about the background, kind of the objectives of this project, and then Dr. Taggart's going to speak to some of the results we saw over the past 2 years. So iron chlorosis is a common problem when soybeans are grown on calcareous soils. These soils are common in the Blackland Prairie area of Mississippi, kind of over here in the northeast down to the eastern part of the state. The severity of this chlorosis has been related to pH, temperature, calcium content in the soil, and then wet soils. So kind of the moisture content of that. Iron becomes less soluble at these higher soil pHs, so that can play a significant factor. But in the past, most of the work in this area has focused mainly on variety selection, looking at less susceptible varieties, kind of doing variety screenings, and on these in these problem areas, most of our producers to date try to plant varieties that may be less susceptible to the iron chlorosis. So our objectives for this project were, one, to determine if 7 selected cropping systems could improve our soybean yields across 6 selected varieties. So with these varieties, we had 3 that were identified with a higher tolerance and 3 with a lower tolerance to iron chlorosis. So within that, we wanted to see if we could implement some cropping systems that were able to improve the soybean yields among these varieties. And our second objective was to evaluate the correlation of soil moisture data with iron chlorosis symptoms across cropping system and variety. So we wanted to see how soil moisture might be affected not only by the cropping system, and then how that might affect the iron chlorosis symptomology we saw throughout the growing season. So moving into kind of the materials and methods here, as I said earlier, we had 6 varieties. We had 3 higher tolerant varieties and 3 lower tolerant varieties. These varieties were selected from screenings that Mississippi State does every year in this area. These varieties were selected in order to kind of represent a lot of the acreage that we see around this part of the state. So in 2019 and 2020, we had two Asgrow varieties, 52X9 and 53X9, two Terrell varieties. The 4927 was the higher tolerant, and 4679X would have been the lower tolerant variety, and then two Delta Grow varieties. In 2019 and 2020, we did have a different higher tolerant variety. Delta Grow discontinued 4670 after 2019, so we moved to a 48X05 variety for the 2020 growing season. So the cropping systems that we talked about, which is what we really kind of wanted to look at, was this project. We had 7 different cropping systems. So we had corn, soybean, stubble at 160,000 seed per acre. This higher seeding rate was selected from some previous literature that I guess looked at seeding rate and how it might affect iron chlorosis, and it suggested that at a higher seeding rate, you may be able to kind of make up for some of that symptomology you may see in the year and protect some of that yield. So that's the reason that 160,000 seeding rate is in there. And then we also had kind of our normal seeding rate at 120,000 also in these cropping systems. So we also had a corn, soybean, stubble plus a small grain cover at 160,000. This small grain cover was selected to be cereal rye. This was also chosen because of some previous literature that said that cover crops may have an effect not only on soil moisture content, but it also may change kind of the chemistry in the soil there with some of the nitrogen content on these small grains, and could possibly play a factor in your iron chlorosis symptomology that you saw throughout the year. The third cropping system was a soybean stubble plus a small grain cover at 160,000. 4, we had the soybean stubble small grain cover plus a roller packer at 160,000. So adding this roller packer in there was kind of to try and try and simulate what you would see in possibly a compaction type scenario. So if you saw the picture a few slides earlier, you could kind of see where there were tire tracks running through this field. And so that is a common scenario that we see in this area. We commonly see tire tracks might show a little bit less symptomology in that. So we think this may be due to compaction, bulk density in those areas. So this roller packer was to simulate that and see how that might affect and play into iron chlorosis and the symptomology. The cropping system 5 was a soybean stubble and a roller packer at 160,000 seed per acre. Cropping system 6 would have kind of been a lot of our common kind of the producer standard here with soybean stubble and a roller packer with 120,000 seeding rate. And then cropping system 7 was simply soybean stubble at 160,000 seed per acre. So this study was conducted at the North Mississippi Research and Extension Center in Verona. It was at the same location in 2019 and 2020. This is a non-irrigated site. All treatments were planted on raised beds with 38-inch row spacing. They were planted on April the 30th in 2019 and May the 13th in 2020 due to the rains. A lot of the iron chlorosis work that you've seen I guess in the past is pushed back to that late window. When we get into those drier and hotter months, iron chlorosis symptomology seems to be greater due to probably moisture availability and then temperature, which I spoke to earlier. This project, we really wanted to look at putting it in those normal planting windows and to see how it would come out there. So we wanted to be in that normal window when a producer might plant to see how it would really affect the producers and how it would play into that. So we had a split plot. So our main plot was the cropping system, and within that we had a subplot was a variety. So each variety was planted in 2 rows in each plot. These plots were 35 by I guess it would be 2 rows of 38-inch row spacing. These were as a randomized block within these main plots of cropping systems. And within that, each treatment was replicated 4 times across the field. So this is kind of how our plots laid out in 2020. So there were 4 replications separated by a 10-foot alley. In 2020, we had corn, which would have been a setup for basically our 2021 plots, as we'll go over next. And then from there, moving basically left to right, we had each of those cropping systems would have encompassed, I believe, 24 rows. These would have been placed within those rows and throughout the replications. In 2021, we will shift these basically to the corn to get into that corn stubble rotation. The oats were planted this year in this method, and then we'll plant the corn basically on the now, I guess it would be the southern side of this plot layout to carry over into 2022 plots. So soil samples were collected for each cropping system and each replicant at our planting. These were analyzed for pH and nutrients. So we had a total of 28 samples there, one per replication. Our watermark 200 soil moisture sensors were installed on June the 12th at depths of 12 and 24 inches. There were 2 sets of sensors in each cropping system. We had 1 set in a high tolerant variety subplot and 1 set in a low tolerant variety subplot. And these were in each cropping system. Our oats were planted on November the 21st in 2019. These were terminated a little bit before planting, about a month before, on March the 16th. And then we planted again this year on November the 6th, 2020. We have a good stand. It's growing now. We will terminate. We'll let that grow as long as we can to get as much biomass out of those cover crops as we can. And then we'll terminate that a little bit before planting time when things start to dry out. Iron chlorosis injury ratings were taken weekly starting from June the 3rd through August the 21st, 2020. So we had 10 total iron chlorosis ratings with this to try and see how those affected maybe iron chlorosis symptomology. These were taken on a scale of 1 to 10, with plants having no chlorosis symptoms rating as a 1 and plots rated with the most severe chlorosis rating as a 10 on that scale. SPAD meter readings, chlorophyll readings, essentially were used to measure chlorophyll content. At first, only in where the moisture sensors were on July 14th, and then July 21st, 28th, and August 5th, 12th, and the 21st, measurements were recorded for all 6 varieties across these cropping systems. So plant height was also taken, and leaf area index were recorded. This was taken in the Asgrow 52X9, the high tolerant, and 53X9, the low tolerant variety, on the same dates that the ratings were recorded. So this was taken a total of 10 times throughout the year in these varieties. And with that, that's kind of the setup of this and with our objectives. And I'll let Dr. Taggart talk a little bit about the results that we saw over these past 2 years.
All right. Well, as he said, this is a group effort, and I'll go over some of the results that we found now. The 2 of us started working on this project, I guess, in 2020, but the first year had been completed in 2019, although we weren't really involved that year. And I'll be presenting data results from both years. So in 2020, you can see that these are the iron deficiency chlorosis ratings, and we see that the corn stubble plots rated a little bit better, and also if you can notice, all the cropping systems that had oats planted seemed to rate a little bit lower. The oat plots were significantly lower than all the other cropping systems. But one thing I wanted to point out, in 2019, the cropping systems that are labeled with corn stubble were actually planted into corn stubble. But then in 2020, we didn't plant the corn in 2019 to have the corn stubble in 2020. So in 2020, these 2 cropping systems right here that say corn stubble were not. They were actually planted into soybean stubble. So for 2020, the 2 cropping systems that have the maroon arrows right here are essentially the same cropping system in 2020. And then the 2 plots with the gray arrow are essentially the same for 2020, although that gets a little more complicated in the next few slides. So here you can see the results for both years, 2019 and 2020. And the letters here indicate our T grouping for the least squared means, and the treatments with the same letter are not significantly different. And I use capital letters for the 2019 results and lowercase letters for 2020. So these are not measuring differences between years, but differences between cropping systems for each year. So I know it gets a little confusing, but I didn't want to have too many slides. So the thing to take away here is that in 2019, the cropping system that rated the best was the soybean stubble at 160,000 planting rate. So here we're looking at the ratings, so that was significantly better. In 2020, we see that that 3 cropping systems actually rated higher in 2020 than in 2019. But the thing to note is in 2020, the cropping systems that seemed to rate better were the ones that, even though there was no corn stubble there, maybe we're getting some residual benefit from that corn stubble 2 years before. And then the cropping systems with the oats planted, bean stubble plus oats, corn stubble and oats, and corn stubble and oats are actually soybean stubble. Those rated better in 2020. So now to look at the results by variety, this is pretty clear in both years. The Terrell 4927X variety rated better as far as symptomology goes. And that was pretty consistent. You can see that the ratings were, you know, you could argue that they might be a little subjective. 2 different people did the ratings in 2019 and 2020, although we see here they are fairly consistent from 2019 to 2020. So now to look at the yield, yield by cropping system. The gray bars are 2019 and the maroon bars are 2020. In both years, the clear yield winner there is the corn stubble at the 160,000 seeding rate in 2019, but also 2022. So remember, in 2020, this soybean stubble at 160,000 seeding rate and the soybean stubble at 160,000 seeding rate are the same treatment in 2020. However, we see a much, you know, we're still maintaining that yield bump. We would assume from the corn stubble that was planted 2 years prior. So both of the plots, the cropping systems with corn stubble yielded better, and then also the cropping systems that had the oat cover crop seemed to yield a little bit better. So the yield comparison by variety, you know, as we said, the Terrell 4927X rated better as far as symptomology, and then it also yielded better. So you don't always see that. Sometimes the cropping systems, even though they might rate a little bit better, they would still yield pretty well. But here we see that the Terrell 4927, it rated well, and we also have the highest, significantly higher yield in the Terrell 4927, and then in both years, 2019 and 2020, and then in 2020, the Terrell 4927 and the Delta Grow 48X05 yielded the best. So some conclusions, in 2019, as we said, planting into the soybean stubble at the 160,000 seeding rate had a significantly lower rating than the other cropping systems. And then in 2020, the lowest ratings were in our 3 cropping systems with oats. Then also in 2019, the Terrell 4927X variety had the lowest ratings. And then in 2020, the same Terrell variety and the Delta Grow 48X05 varieties had the lowest average iron deficiency chlorosis ratings. And then also, in 2020, even though the taro variety had was the best yield, the Delta Grow 48X05 was number 2 behind that. In 2019, the corn stubble cropping system planted at 160,000 seeding rate produced significantly higher yields than the other cropping systems, even though 3 other cropping systems had lower ratings. So in 2020, one of the 2 soybean stubble plots had the best yield, followed by the cropping system with the oats. And also the taro 4927X was the variety with the best yield in both years. And also those varieties generally followed the same trends pretty closely in both years. So, a couple of things we're left scratching our heads a little bit. It does appear that, in 2020, we're definitely getting some residual benefit, we believe, from the corn stubble in those plots. So that'll be real interesting this year. Those plots were planted in the same location, but this year we'll be shifting them down to 2 plots. So that'll be interesting to see that. And then as far as the oats go, it's hard to say at this point if the oats, one of the theory with a cover crop is that they're uptaking some of those nitrates, which the benefits coming from that reduced nitrate in the soil. However, it could also be from a little bit of benefit from soil moisture. Those oats might be helping retain more soil moisture in the soil. So, it's hard, in a non-irrigated study, we don't really know. So it'll be fun, I think, to dig into more into the oats and see. We do have the soil moisture data, although we have not really compared that to the cropping system, although that should help us answer some of those questions with the oats. And I think that's all. I just want to say thank you to the Soybean Promotion Board and all the growers who helped fund this work, and also to the Verona crew and the student workers who helped us. As I mentioned, it was definitely a group effort. And then here is the scan code if you're interested in getting those continuing education credits. You should just be able to hold your phone up and click on your camera and hold your camera over the scan code, and it should take you to the correct link. And I think that concludes our talk, unless Justin's got anything else to add.
Excellent. Well, thank you guys very much. Those are incredibly interesting presentations with a lot of questions we were fielding in the Q&A and in the chat. And we just want to encourage anybody who's still has a last minute question, you're welcome to put them there, or certainly to reach out to our speakers for today. We are going to go ahead and Dennis, do you want to bring everything to a close?
Yeah, thank you, Michael, and thank you to the Mississippi farmers for the soybean checkoff funds. You just got a small glimpse of our scientists this morning, and you can see the outstanding research we're doing in Mississippi. And we're fortunate to have such a good group here in Mississippi. And we're fortunate to have the soybean checkoff dollars. If you have any questions and have any interest in some projects in the future, make sure you contact us, and so we can address those issues. And continue to follow mssoy.org. Continue to look at our monthly article in the Delta Farm Press. That's a very impressive article we're doing, and continue to get the latest from the Mississippi Soybean Promotion Board. We thank all of you for joining. We also thank the Mississippi Delta Community College. They have 2 classes joining us today for this live broadcast. And it's always great educating those young guys back home. So thank you all for joining, And we'll see you next time on Research Roundup, and I'll turn it over to Michael.
Thank you very much for bringing that to an end. We will be sending out the recording to everyone who's participated as well as signed up. Make sure you come back and visit the website for any more details. And if you have questions for our speakers, please feel free to reach out. All right, have a great Thursday, everybody, and we look forward to seeing you next time. Take care.